Anyone give me a good definition for chromatography? Where do you think that word comes from? If you think about the parts of it. Anyone know what chroma means? Any Latin scholars?
Isn't it like color or something?
It is, exactly. Yeah, chroma just means color. And tography basically means like the study of. So it's the study of color, which was the original way that scientists figured out how to separate things, was by separating colors, seeing that you could take something that was green and separate it into something that was blue and yellow, take something purple and separate it into something that was blue and red. So you're going to do exactly that kind of experiment tonight. So what we're going to learn about is the ability to separate mixtures, and by taking advantage of difference in their physical properties. So I did a ton of chromatography when I was at UCLA. It was a different sort of chromatography though. We would flow mixtures of proteins and peptides over a column at extremely high pressure and separate them that way, based on these differences in physical properties. So some were more positively charged than others. Some were more negatively charged than others. Some like to be in water solvents rather than hydrophobic solvents. And so you take advantage of that in order to separate, take a really complex mixture and separate it into its component parts. So what we're going to do with this particular lab is we're going to do paper chromatography, which is like the original chromatography that was discovered. And what that means is we are going to separate different compounds by moving them across a strip of paper. Now, the difference in physical properties we're going to be looking at is some of the compounds are going to want to bind to the paper, and some of the compounds would rather be bound to the solvent we're going to use that will wick up and move through the paper. I'll talk a little bit more about that in a minute. So we're going to produce chromatograms, which is just another name for a physical separation of something. We have 3 dyes, red, blue, and yellow. I'm also going to do green. But anyone know what FD and C stand for? Think of something called the FDA.
Food and drug.
Mm-hmm. So you figured out what F and D stands for. What about C?
Color.
No,
cosmetics?
good guess though. Cosmetics, excellent. Yeah, food, drug, and cosmetics. So these are dyes that have been approved for you to either eat or put on your body in some way. So, you know, they are not harmful. So they can be put into Kool-Aid for kids and stuff like that. So we're going to see how they separate on the paper, and then we're going to compare them to the same thing for 3 Kool-Aid samples, orange, grape, and strawberry. And so we're going to see if we get the same pattern of spots with the Kool-Aid samples that we do with the food dyes. And will we be able to identify the dyes in the Kool-Aid based on the pattern of spots we see from the known dyes. So let's just go over a little bit about how that's going to work. So as I mentioned, we're going to be using something called paper chromatography, which is exactly what it means. It's just, yeah, this happened last night too. Ah. There we go. So what we're going to be doing is taking advantage of the fact, as I mentioned, some of these dyes are going to want to bind to the paper. And some of the other ones would rather be bound to the solvent. Now, anyone tell me what paper is made out of?
Like wood fibers?
Mm-hmm. That's right. And what are the wood fibers made out of?
Cellulose?
Cellulose, right. Cellulose. And do you know what cellulose is?
That's just plant matter, right?
But do you know what it's made out of? Like this, it's sort of based
Cells?
on a fairly common molecule.
Sugar.
Sugar, that's right. Basically, it's just a polymer, sugar. That's why termites can basically devour your entire house and have like a full belly, because they, in their gut, have an enzyme that can break down the cellulose into common sugars. So for them, it's just like looking at a big sugar cube, a piece of wood. They can just like eat through it. If we eat cellulose or paper, we do not have the enzyme that can break that bond to liberate the sugar. So we don't get any calories out of it. It just passes right through us. So sugar, I think we looked at what sugar looks like a little earlier. The simplest sugar, like glucose, has 6 carbons, and attached to each one of them is something called a hydroxyl group. And when we do organic chemistry, we'll talk a little bit more about them. But OH minus, we know is a hydroxide ion. And when it's bound to carbon, we call it a hydroxyl group. So sugar has all these hydroxyl groups on it. And we know that water has an O and 2 H's on the side. And we know that that's extremely polar, right? That is a very polar, one of the most polar molecules we have. And so sugar is also extremely polar because it has all these oxygens with hydrogen on one side and carbon on the other. And we know that oxygen is a lot more electronegative than either carbon or hydrogen. So all these oxygens have a slightly negative charge, just like the oxygen in water, while the hydrogens have a slightly positive charge. This is why sugar dissolves so easily in water, because they're both really, really polar. And so they fit together really well. The slightly negative parts of water and slightly positive parts of water bind to slightly negative parts of the sugar, and they just get along just great. So this is a very, very polar molecule. So the very, very polar dyes are going to want to interact with these groups on the paper. And so as the water moves up the paper by capillary action, we'll talk a little bit about what that means in a second. As the water moves slowly up the paper, those dyes that would rather be bound to the paper are going to be retarded in their progress. They're not going to move as fast. On the other hand, our solvent is going to be water with some sodium chloride in it. Now, what kind of compound is sodium chloride? Is it a polar, non-polar, or ionic compound?
Ionic.
It's ionic, right? So I've got a polar surface, the paper on one side, and moving up it is an ionic mixture. Some of these dyes actually have a charge in them. Some of them are going to have a positive charge in them. Some of them are going to have a negative charge in them. So they're going to be, have some, easy for me to say, they're going to have some ionic character. And so those dyes that have more ionic character than polar character are going to be carried along by the solvent. And so as the solvent moves up the paper, the molecules with ionic character are going to move with the solvent, and they're going to be pulled up the paper higher. And that is where our separation comes from. The more polar molecules won't move as high up the paper, and the more ionic molecules will move higher up in the paper, depending on whether they'd rather be bound to the polar paper or the ionic solvent. Okay, does that make sense to everybody? Hopefully.
I thought the water, you know, since it has the hydrogen and the oxygen, wouldn't it be also ionic too?
No, water is polar, it's not ionic.
Oh, okay.
Yeah, it's not ionic. To be ionic, you have to have a charge, remember?
Right.
So the water is going to have charged molecules in it. We're going to put sodium chloride in it, meaning that the water won't just be polar, it'll also have ionic groups in it. So it'll be even more polar than usual. It'll be ionic. So let's have a look and see what all these different terms mean when we talk about chromatography. So here is a setup. It's a little more fancy setup than we're going to be using, but basically the same thing. We're going to have a chamber. In our case, we're using a test tube. We're going to mark a line across our paper that we're going to call either the baseline or the origin. That is where we're going to spot our dyes. So before we begin, we're going to take our dyes and our Kool-Aid, and we're going to put a spot of them right there in the middle of the baseline. Okay, that's where we're going to spot them. And then we're going to let them dry for a minute, and then we're going to put them in the test tube. So the paper we call the stationary phase. Why do you think we call it the stationary phase?
It does not move.
It doesn't move, exactly. Yeah, you'd have to think, don't think too hard. Some of these questions aren't as tricky as you think. That's exactly right. It's a stationary phase because it is stationary. When we put it in the test tube, it doesn't move. On the other hand, the solvent that we put that paper in does move. As soon as we put it in, it starts wicking up the paper, and then through capillary action, it starts to move its way up the paper. Does anyone know why water, what does capillary action mean anyway? When I say that, what does that mean to you? Have you heard that term before? You must have heard capillary action before.
Well, it would describe how water moves from the roots of a tree up to the leaves and to the top, or how blood moves through your capillaries in your body.
Exactly. Do you know why it does that, or how it can do that?
A pressure gradient, I think.
Yes, but it's more based on the fact that water is so polar that, so we have our slightly positive, slightly negative, that water is so polar, it basically forms a network of all of these waters bound to each other. That's very, very strong. It's a very, very strong interaction. It's one of the reasons why it takes so much heat to boil water, because liquid water is bound together by itself, and it takes a lot of energy to break those interactions. And the same thing with capillary action. If we have water here and it gets moved, this water molecule moves up just a little bit, it basically pulls all the other water molecules below it along with it because of this interaction. It's why redwoods can grow to be like over 1,000 feet tall, because this interaction is so strong that water evaporating from the top creates that pressure gradient you mentioned that moves another water molecule up, which pulls that whole column of water up. And so as the water is moving its way to the top to evaporate, it pulls, well, that looks like some Martian language. It pulls all the rest of the water up along with it. And so, as I mentioned, those compounds that most want to be bound, that are most bound up with the solvent, which is water with sodium chloride in it, so it's ionic. Those compounds that are more ionic are going to move further up the paper because they're going to be spending more time interacting with the mobile phase. The mobile phase being the solvent, and mobile because it's actually physically moving up the paper. Those compounds that are more polar than ionic are going to be down here. They're only going to be moving up a little bit because they're spending most of their time interacting with the paper, not the solvent. It isn't that they're not interacting with the solvent at all. There's always a little bit of interaction back and forth, but most of the time they're spending most of the time interacting with the paper. And so they won't move as high. And so we can say that the more ionic compounds move to the top and the more polar compounds only move a little bit, and we'll find them at the bottom. So how are we going to measure this? I mean, we're physically going to measure it because we're going to measure the distance from the baseline here and measure how far the spot moves. And we're going to measure how far the solvent moves. So we're going to measure those. That's part of your lab report. We'll be measuring those distances in millimeters. So how far up the paper did they move in millimeters? But how do we, what is the, are we just going to be done with that? Like just measuring in millimeters? Is that, will we use that measurement only to determine whether we have the right dyes in the Kool-Aid or not? Is it just the distance they move that we're going to measure? Or is there something else we're going to calculate? You can see right on the slide.
Retention factor.
So what is that going to, what first of all, how do we calculate that? Oh yeah, what is our solute? When I say, what does solute mean? Anyone know what solute means? If you have a solution, you have two things. You have a solvent and you have a solute. What's the solvent? We know what that is. What's the solute?
It's what's dissolved in the solvent.
Exactly. So in this instance, our solutes are going to be the different dyes that are going to dissolve in the solvent and then start moving. So we're going to calculate these distances. So the distance the solute travels are these distances here. 1, 2, 3, 4. And we're going to measure them from where they started. Excuse me. We're going to start them at the baseline, and we're just going to measure how far they moved in millimeters. So what we're going to try and do is find the middle of each of these spots. And these spots are going to be, some of them are going to be like really big, you know, like that. We're going to try and find what's is the densest part of that spot, and then find the center of it, and then mark it with an X. So we'll see how far each of them move from the baseline. And then we're going to measure how far did the water plus sodium chloride move. That'll be a little easier to do when you take it out of the test tube. It's kind of hard to see because basically you've just got a separation of dry paper and wet paper, which is a little hard to see. I'll show you that an easy way to see it is if you backlight it with either a lamp or your phone or something. If you just put the backlight against it, it's much easier to see where the solvent front is. But why do we bother calculating this retention factor? Why don't we just measure the distance and then compare the distance on one chromatogram to the distance on another one? Why bother with this? Like if I do this experiment and my red spot moves, 46 millimeters, and Eva does the same experiment and her red spot moves 49 millimeters. Well, that's close enough, right? We can say that's the same, it's the same thing, but we don't. We do this calculation. Why do we bother doing that?
Eliminate errors, maybe?
What errors? It's not really an error. We're trying to eliminate variability. What variability do you think we're trying to eliminate?
How quick the solute travels?
Right.
Or is it more because when we measure it, it might be a little bit off?
There's that. That's part of it. But there's certain ways that like Eva and I could do the experiment that might be different. What are some of the ways that we could do the same experiment but do it a little differently?
Use smaller cups? I mean, like shorter cups.
Not so
Maybe
much
I'll
the cup, but what if our
influence-
paper's not the same size? What if the paper she uses is twice as long as the paper I use? Can we directly compare the distance the spots move then?
No, because wouldn't it be longer or greater solute on the paper versus less solute on the paper, which would affect the result?
No, but on the longer paper, she would probably let hers run a lot longer because it would take, if her paper is twice as tall, it would probably take twice as long for the solvent to move up the paper. So if she runs her experiment for 30 minutes and I run my experiment for 15 minutes, her solvent and her spots are going to move twice as far as mine. So we won't be able to directly compare distance, but the ratio of how far her dye spots move versus the solvent should be exactly the same as mine. If we both do the experiment carefully, the ratio of how far the solvent moves to how far the spots move should be the same regardless of how big the paper is or how long you let it run, because that ratio stays this. The ratio is a constant. The distance isn't. It's sort of like density. The mass can change and the volume can change, but the ratio between them is always the same, provided you have the same substance. So if I want to compare these spots, let's say that's my Kool-Aid. If I want to compare these spots to the dyes, which are like these standard spots, standards meaning I know what they are, and I'm basically measuring how they behave. I want to compare known things to unknown things. So if I'm comparing my standard dyes to the Kool-Aid spots, what two things have to be the same for me to call them the same dye? When I'm comparing the chromatograms of one of the Kool-Aid chromatograms and my standard dye chromatograms, what two things have to be the same for me to say that's the same dye in both?
You have the same retention factor?
Yes, that is one. Absolutely right. One, the RFs should be similar. I mean, we're not dealing with that. It should be similar. We're not dealing with, you know, super accurate measurements here. So like I'm saying, within 20% or so, yeah, we'll say close enough. So that's one. Yes, their RFs have to be similar. What's the other one? Don't think too hard. So I'm comparing my standard dyes. I'm seeing how far they moved up relatively to the solvent. I'm looking at calculating the RFs, and I'm also looking at my chromatograms of the spots of the Kool-Aid. So I've calculated my RFs, and they look fairly similar. What's the other thing that I'm looking for that should be the same? Don't overthink it.
Their physical appearance?
That's right. And so what's a simpler way of putting their physical appearance?
Color.
Their color, exactly. Excellent. Their color should be the same. Because if I measure two dyes with the same retention factor, let's say I measure my red dye, my standard dye has a retention factor of, let's say, 0.50. And the green or the yellow dye on one of the Kool-Aids also has a retention factor of 0.50. I can't say those are the same dyes, can I? No, because they're different colors. So they have to have both things in common. The color has to be the same and the retention factor has to be similar. Only then can I say that those are, like those are the same thing. So we have to agree on both. Excellent. Okay. So there's a lot of stuff to dig out. So hopefully you found the micro tip. There's a couple of things that are going to be tricky to find. One are the micro tip pipettes. They're different from the typical ones we've been using. They have super fine ends on them. I think there's 6 of them in there. So those aren't the regular pipettes that have a slightly wider bottom. They're very fine. The other thing are the pins. There's, I think, a bag of 8 pins, and they come in a bag that I think that was taped to the food dye kit. There's like a kit with 4 food dyes in it, and the pins were taped to that. So hopefully you found that. And then these are the polystyrene test tubes that we used last week. Hopefully you've cleaned them. Now the chromatography strips, I want to talk about those because of what we did last night. So I'll go over that on the next slide. Has everyone found those papers? I think they're in the same bag with the Kool-Aid and the microtip pipettes. Has everyone found them? See?
Yes.
Yes?
Yes.
Okay, good. So a little bit of foil or something, just something you can put a drop of food coloring on that won't stain everything. Something that'll just beat up. And you can then dip the tip of the microtip pipette into it and get a little out of it. Aluminum foil works pretty well, or just a piece of plastic will work well. Something to stir the Kool-Aid up. And at the end, the third thing you're going to be doing, so you're going to be comparing the standard dyes. You're going to be looking for those dyes in the Kool-Aid. And the third thing is finding something in your house to also do a separation on. And so so M&Ms are pretty good. Somebody else said that, what are the really hard chewy candies that aren't M&Ms?
Skittles.
Oh, the colored Mentos that'd work too. Colored, but I'm thinking of the fruit flavored, and they have the hard shell on them. They're super yummy. Skittles, that's it. Yeah, Skittles would work awesome. And if you don't have that, just some sort of like a black marker works really well. Black marker works really well, actually, because if you do a chromatogram on a black marker, you will find that all the colors are in there. And that's how black is made, right? You basically mix all the colors together and it makes black. And so that works really well if you have a black marker. So
You said that's an alternative to the candy?
Yeah.
You said?
mhm.
Okay.
Yeah, it's not every, some of us are, watching our girlish figure. It may not have a lot of these things, around the
I
house.
found leftover M&Ms from my daughter, but if I don't have to take them from her, then I'll use a black marker. I want to make sure.
Perfect. So to prepare making the solutions is really, really easy. So just weigh out 2 grams from each of the Kool-Aid packets. And then, so you have the small cups, the really little ones, and mix them with like 5 mils of water. So they're going to be super concentrated. So don't drink them because they're really, really, they don't have any sugar in them. So they really taste awful. So for our mobile phase, our solvent is just 100 mils of water with 0.1 grams of sodium chloride in it. So hardly any, like just a tiny bit of salt. So 0.1 grams. So notice it just says 0.1. So anything like from 0.7 to like 1.4 is fine, so that'll give you a 0.1% solution. So now I sort of want to mention the chromatography strips. So you have 6 of them, right? And what they recommend you do is to fold them in half and cut them. And what we found last night was, at least for the standard dyes, that may not work particularly well. So what I'm going to suggest is that you take 3 of them and cut them in half exactly the way it's shown here. And the other 3, just cut a little bit off the side, about 5 millimeters or so, just a tiny bit. It doesn't have to be super precise, just enough so it fits in the test tube. Okay, and then we're going to use that for our standards, because we noticed that the standards, since they're so concentrated, that we tried spotting them several times last night. We tried spotting them with like 1, 2, 3 spots. It seems that just one application of the food dyes is more than enough. So if you get like a really small spot, that will work really well, because most of us found that the food dyes spread all over the place and made it really, really difficult to measure the RF. So I think what will make that easier is if we have a wider strip. So this is what the strip looks like when you cut it in half. So these seem to work just fine for the Kool-Aid. And these are the ones we're going to use for the food dyes. okay? And we'll measure them exactly the same way. So we go 1.5 centimeters from the bottom, and we draw a line across it. That is going to be our origin. And we'll spot our food dye right in the middle of it. It also helps to cut out to make this little V at the bottom. And the reason for that is we're just going to put a tiny bit of solvent in the bottom of the test tube, only like about 1 ml or so. And so this little point here will help the water wick up and actually move in a straight line. If this was flat, then we could have some of the water come in from this side or some of the water come in from this side, and our solvent front might be a little crooked. But if it only comes in at one point on the bottom, we get a nice straight solvent front. So that works actually really well. So make sure you do that. So the other kind of thing that's a little confusing, the way this is written here, is this 9.4 centimeters is from the bottom, not from the 1.5 centimeters. So we go 1.5 to the origin, and then we go like about another 9.5, and we put another line across. This is where we're going to stick our pin. We're going to put our pin right in the middle, right through the paper. And what the pin is going to do is hold our paper in place and keep it from hitting the bottom of the test tube. So we want the tip to just sit in the solvent. We don't want it to hit the bottom. So these dimensions are going to help us do that, okay? Now, your 6 microtip pipettes, measure them about 5 centimeters from the end, and we're actually going to physically cut them with a pair of scissors, because we don't want to actually, the spots we want to put on these papers are so small that we don't actually want to draw any liquid into the bulb. That would be way too much. We're just going to let capillary action pull a tiny amount of liquid up into the tip, and then we're going to spot it. So our spot should be really, really small. So any questions about the paper? So 3 of them, cut them in half, just like the instructions say. And the other 3 of them, just trim a little bit off the side, about 0.5 millimeter or 0.5 centimeter or so.
Is there an issue if I ended up cutting all 12 lengthwise already?
Oh, that's fine. Okay. That's fine. Yeah, this was just something, yeah, I mean, I did the same thing. I did the same thing last night. I had like 6, I cut up 3 of them, so I had 6 of these. And then I figured I would try a wider one, and it worked a lot better. So I wonder why they just don't give us more. It's just paper. I don't know why we had to have only 6 strips of paper. They should have given us like 20 of them or something. It's not, I mean, you could use a coffee filter. So if you want to, if your experiment tonight doesn't work, you could just use a coffee filter, and it's the same paper basically. So we're not dealing with, you know, super expensive materials.
So I just want to make sure the total length of the paper is 9.4 that you have to measure centimeters.
No, the paper will be larger than that.
Oh, well, I mean the lines you have to draw.
Yes. So 9.4 from the bottom.
Okay.
And 1.5 from the bottom.
Okay.
Yeah, I don't like the way they.
Yeah. I was
Got
like,
that there.
wait, do you measure 1.5 and then you add 9.4 so
That's what it
total will be
looks like,
11?
right?
Yeah.
That's exactly what it looks like. So yeah, that part's confusing. That's why I wanted to make sure I explained that one. So for the known dyes, all we're going to do is just spot once. Okay, on the middle of each one of these. So we're going to take our red, and we're going to put one spot right there. And then for the blue, we're going to put one spot right there. And the smaller, the better. because like I said, these things are going to spread like crazy. So then we put about 1 ml of the solution in the bottom of the test tubes. And we're basically going to watch and see what we're looking for is we're going to watch the dyes move. And if you want to know where the solvent front is, like I said, the easiest thing to do, you'd either pull it out by the pin and look, or you can put a light up against it. The wet paper should look a little brighter than the dry paper. So you should see like this sort of brightness moving up the paper. So the way we're going to measure these, so that you're going to have a tiny spot here, and then believe me, you're going to have like a huge spot up here. So it's going to look, to have color all over the place. So what you want to do is find the densest, the darkest part of the spot. Let's say the darkest part of the spot is right there. And then put an X right in the middle. Wait till it dries and put an X right in the middle. And then we're going to measure the distance from where that X crosses and the origin. Because basically what we want to do is, since the middle of our spot was here, we want to find where the middle of our spot is here and then calculate the distance. Because if we try and measure the top of this thing, that's going to be wider. That will have spread out. We want the middle, the distance from the middle to the distance to the middle, basically. So that will be our D spot. So that you wait till it dries before marking it. What you do have to draw while it's still wet is the solvent. So if the solvent goes up to there, when you pull it out, lay it down on some paper towel, and then carefully draw where the solvent front was with a pencil. Why don't we use a pen? We're going to make all these markings with pencil. This is going to be marked with pencil. This is going to be marked with pencil. We're going to mark the solvent front with pencil. Why a pencil?
So the dyes don't spread from the pen.
That's right. Yeah, pen is all dyes, right? Especially if you have a black pen that has all the colors in it. And so if that gets wet, that just starts spreading out and making a mess. So the pencil, the graphite stays put. It doesn't move. Graphite isn't particularly what we say hydrophilic. It isn't water loving. And so it's not going to move when it gets wet. So then we measure the distance of the solvent. We can do that as soon as possible. And then once our spot dries, we can determine where the center of the spot is, okay? So I think a single spot is probably good enough. I think one of the mistakes we made last time was we put too much dye on it. The blue will be really easy to see. The red will be really easy to see. The yellow might be a little tricky because the yellow against white might be a little harder to see, but I think a single spot is probably better than having them spread out too much. So this is where we put our data. So what color is the band? Well, gee, I wonder what color the red's going to be. And then you measure distance the band. So this would be our D spot in millimeters. And this would be our D solvent in millimeters. And then this will be the ratio, spot over solvent. What is going to be the units for RF, for our retention factor?
No unit?
Excuse me?
No unit?
No unit. That's right. Very good. Because we're going to have millimeters divided by millimeters. And as we've learned from doing all of our unit conversions, if you have the same unit in the numerator and the denominator, they cancel. Right. So I'm always yelling at you, and I will be yelling at you on your test, when you leave units out of your answer. This will be the only time that I will yell at you if you put units in your answer, because RF is a unitless number because it's a ratio. And that happens a lot in science. There's a lot of measurements we take that are ratios of the same thing. And when we do that, it just has a ratio. So no actual unit, just a number. Good answer. Yeah, so we put our known dyes in here, and then the unknowns for our different Kool-Aid flavors in here. Now, I think on the actual report template, there's room for only 2 bands. If you have more than 2 bands, you can add another column. But I think mostly these things separate into either just one or 2 colors. So if your band has the same color and the same retention factor, you can identify it. If either the retention factor is the same, but the color is different, or if the color is the same, but the retention factor is different, then you can't identify it. And that's fine. We can't always, identify things without any error. So only identify it if both of those things agree. And like I said, the RF should be probably within around 20%, maybe even 25% of each other. If it's like 50% different, 60 versus 0.6 versus 0.4, then yeah, that's a bit far off to say it's the same thing. Okay. And then the last one, yeah, just do whatever you want as far as the third activity goes. You can use, if you have some other kind of like powder for making some sort of drink, like Gatorade or something, you can try that. Or the M&M, something with a hard color shell on it, or pen if you have nothing else. That works really well too. Okay, so everyone's going to get all their stuff together.
So, Professor, for
Yes.
the last part, because I only have 6 of the paper.
Right, yes, so cut 3 of them in half and leave the other 3, just trim the other 3 a little bit, leave them larger. And
The
we'll use-
larger one would be for the dice, right? And then the other ones can be half. Okay, thank you.
Yeah, the other 3 you cut, then you'll have 6 pieces. You use 3 of them for the Kool-Aid, and the other 3 you can use for whatever you like.
Okay, all right.
Yeah.
Thanks.
All right, I'm gonna get my stuff.
Professor, I have a question.
Yeah.
If you only have sea salt, does it make a big difference or?
It shouldn't. It shouldn't. I mean, sea salt has potassium in it too, but it won't matter for this.
Okay.
Because basically we're looking at physical interactions rather than chemical. There are some chemical reactions where potassium would screw things up because it's sufficiently different from sodium in size. Sodium is a lot smaller than potassium. But since this is a physical reaction, it shouldn't really matter. So no problem. Yeah, I've heard that a lot, that like some people don't have pedestrian salt in their house.
Will we need it in future labs though? Because then I'm gonna have to get some.
Oh, that's a good question. I'm not sure. Let me check on that. Yeah, because it is sort of assumed that everybody has table salt, and this is not true. Yeah, someone wanted to use pink Himalayan salt, and it's like,
I was just about to
don't
ask
waste that on a chemical experiment.
So that wouldn't be any good to use pink Himalayan.
No, for this one it'll be fine. But I mean, you're probably going to have trouble measuring 0.1 grams of it, because usually, I mean, it's some pretty big crystals, right? So
Yeah, it's a little bit bigger. I do have kosher salt, but that's even bigger.
That's even bigger. Well, you can take some kosher salt and just crush it a little bit. That'll be fine. Because remember, you don't have to be super accurate with this, since it says 0.1 grams. I mean, if it's 0.2 grams, it probably won't matter. We won't tell the company that, but it probably won't matter.
Professor, the strip that we're cutting to make it skinnier, is that the half strips or the full strips?
We're cutting the full strips to make half strips. I don't
No, 'cause
understand
you
the
were saying
question.
some of them have to be skinnier to fit in the test tube.
Oh, so the full size, the ones you have right now with nothing, you've done nothing to them, are just slightly too big to fit in the test tube. So I recommend you take 3 of those and trim a little bit off the side so that they fit in.
Got
That
it.
means you'll have 3 of the original larger ones left. Those, you cut those in half.
Got it.
Okay, yeah.
I was wondering, I mixed the Kool-Aid with a bigger cup. Is that gonna be an issue?
Mixed it with what?
Oh, well, I used the bigger plastic cups.
Oh, it doesn't
Instead of
matter
the
what the
small
size
one.
of the cup
Oh,
is.
okay.
Just have make 2 grams of the Kool-Aid with
Right, yeah, I
5
do.
mils of water.
Yeah.
It really doesn't matter what size thing you put it in. It just may be a little tricky to reach down in there and take a spot. But, you know,
Oh, I thought we were gonna use a pipette to just drop a spot in the paper.
no, remember we're cutting off the tips of the micro pipettes.
Oh,
Let me just grab one. All right, so these are the tips. We just want the tip of the micro pipette. You can see how small that is. So basically, we're just going to dip that into our dye or Kool-Aid, anything we have the colored dyes in, and just have a little bit of it. Don't squeeze it. Don't do anything. Just allow capillary action to pull up just a tiny bit of liquid. And then we just move it over to the paper and then just spot it right in the middle. We want as small a spot as possible, because you can see what happens when we have a big spot. You can see how this is one we did last night. You can see how much this spread out, right? And this is actually one of the better ones. This spread out a lot. So this is our blue. And the red one seems to spread out even more than that. So then when we tried a bigger piece of paper in just a single spot, this was the green food dye. And you can see this separated beautifully. It didn't spread out nearly as much. And we could easily find the middle of the blue and the middle of the yellow. So this was green at the beginning, and then it separated really nicely into blue and yellow. So, and I think the secret was to not have, most of us like spot it more than once. So I think spotting just once is probably better. So when you so this is about how much liquid you're going to want in the bottom of the test tube. Not very much, about 1 ml or so. So when you put your paper in, the tip of it only should just it shouldn't hit the bottom of the tube. It just should touch the solvent. And then it'll start moving up. You also want to avoid having the paper lean against the side like this. You don't want it to lean against the side. You want it to, that's why we use the pin, so that it doesn't stick to the side of the tube. Because if that happens, all of the solvent will just rush up the sides, and you'll just get a big mess, even bigger than this. Yeah, so then you should have a nice little point like that. And that should almost reach the bottom of the test tube, but not quite. So some students last night had an issue with they didn't have these micro pipettes. Some people were given just the regular ones, which you do not want to use for this. And if that's the case, sort of did a little workaround. Whereas if you take the pin and dip the top of it in the dye and then quickly just spot the paper, you get a really nice round small spot. And that seemed to work for some folks. So what do you know? So I think I'm going to try that my own self.
For the other measurement, the 9.4 centimeters, that's where
That's where the
It's just-
pin is going to go.
it's just a line, but what we're trimming is from the bottom to 1.5 centimeters, right? Like for the
Yes,
for the
yes,
point.
for this.
Yeah.
Yeah, so that's where the trim, so that's 1.5 from the point. And then
Okay.
we don't cut anything on the top. So this is the line 9.4. We don't cut that at all. We don't do anything to that. That's just where our pin is going to go. So on the top of this, you could actually write what you're going to spot. You can write, blue dye, red dye, yellow dye, grape Kool-Aid, whatever. That's probably a good idea to do that so you don't mix them up. In fact, I'm going to do that now while I'm thinking about it. How you doing, Ricky?
Pretty good.
But it looks like you're playing trance tonight, or is it a house? What kind of music you DJ in there?
Oh no, I'm just used to hooking up my audio through my headphones, so I never use my monitor's audio. So.
Okay. All right, let's see how we do here. Yeah, so this was a spot I made with the point of that's probably even a little bit bigger than you want. If you can get it a little bit smaller than that, that's why I think the micro pipette is the best way to go. Because yeah, I saw some people made some really nice small spots with the micro pipette last night. Because this is probably going to spread. So I let this sit for a minute and work its way into the paper. Because if I don't, it's just going to dissolve into the solvent, right? And we don't want that. We also don't want the solvent to come up to the dot. How come we don't? Why don't I just dip the whole thing in water? Why don't we just fill the test tube up with water and salt? So I definitely do not want the solvent to come up to here. I just want the solvent to touch the bottom. Why is that so important?
It might dilute the solvent. The solute, I mean.
Yeah, exactly. We want the solute. The only way we want the solute interacting with the solvent is on the paper. Because even though, it may be ionic or polar or whatever it is, it's going to dissolve in water because pretty much everything does. So the only way we want it to interact with the water is if by coming through the paper. We don't want it touching the water directly. Yeah, so then you just stick the pin right through that line that you drew at 9.4 centimeters. And we've got that little bit of water and salt in the bottom, about a mil or so. Then when we put it in, we want to make sure it doesn't touch the sides of the tube, and we don't want it to touch the bottom. So that's perfect. It doesn't touch the bottom. And you'll see the water moves really fast. This experiment only takes about 10 minutes to do, so this doesn't take very long. The water has already gotten to the dye. It's already got to the dye. So it moves really quick. So keep your eye on it. You do not want the solvent to go past the pin. Because if the solvent goes all the way to the top, what happens to the spots? If we were to leave this overnight, what would happen to all of the spots? What do we think would happen? Oh, there it goes. You can see it's separating already. So if I was to leave that overnight, what would happen? I got yellow separating from blue here. So if I left it overnight and came down the next morning, what would happen? Where would my spots be? Keep forgetting this is the quiet group.
Would it continue bleeding off to the end of the paper?
Exactly. Excellent, Liesel. That's exactly Liesel. I'm always getting it wrong.
Liesel.
See, I told you. Yeah, exactly. That's what happened. They're just going to keep moving, right? So the solvent is just going to get to the top and then evaporate. And then the solvent's just going to keep moving, and it's just going to keep pushing the spots up higher and higher till basically they all just get to the top. So eventually, the retention factors for all your spots will be 1, because the solvent will have gone to the top and your spots will have gone to the top too. So basically they will have separated and then rejoined as they sort of pile up on the top. So we don't want that to happen. So stop it about a centimeter or so before it gets to the pin. That'll give you plenty of separation.
In the beginning, when we place the 2 drops of the dye, it's the dye we mixed,
Just one.
right?
Just one.
Oh, it says 2.
Yeah, but I said 1.
Okay.
So we're just going to do 1 because
And we just drop it on
a
the-
really small spot.
And we drop it on the aluminum foil, right? Before we put it on the paper.
Yes. Yeah, because that just 1 drop of food dye is about 1,000 times more than you need. So you take I put mine in little tray here. And so you can see, I mean, I've got a ton of it left. So yeah, you want it on something that'll just stay put. So the surface tension of the dye will just keep it together as a spot. And then you take a tiny amount out and spot it. You're only going to need 1 drop.
So should I do it on the aluminum foil or on the paper towel? Because it said,
Well, if you put the dye in the paper towel, it's just going to spread into the paper towel. You want to
Oh.
spot it on something where it won't move. So aluminum foil or plastic wrap or one of your way boats,
okay.
something plastic that it's not going to seep into.
And you just use the regular pipette to get the drop, or do you use the micro pipette?
No, these will just drop by themselves.
Oh, I thought they wanted us to use the prepared Kool-Aid ones.
Yeah, we're doing both. We're doing 6 of these, and then you're doing another one. We're doing the 3 dyes, 3 food dyes, and the 3 Kool-Aids. That's 6, and then the 7th one is a wild card, whatever it is you want to do.
Okay.
So we're spotting on the chromatography paper with the micro pipette. So I wasn't quite, we're not using the micro pipette to put it on the foil. We can just squeeze a drop out of this to put on the foil. Was that what you
Okay.
were asking? I was confused.
Yeah.
Okay. So this is only about, I don't know, 4 minutes in, and you can see how well the green is separate. I don't know why they don't have us do the green, because the green gives you a really nice separation. So if you want to do the green, feel free. It gives you a nice separation.
Did I need to wear my glasses? Oh, fogged up it is. Red, yellow, blue. Did you want the food dye to all mix together? They said one on top of each other.
No. On separate pieces of paper.
Oh, okay, because it said a drop of each on top of each other. So that's why I thought maybe you wanted to mix them.
Where does it say that?
On 3 separate locations on the plastic wrapper aluminum foil, place, well, 2 drops, you said 1 drop of each of the dyes
Right.
on top of each other from each dropper bottle.
Wow. They really don't know how to word things, do they?
So I thought, you know,
No, we don't want to
you
mix
mix
them.
them.
No. Although you can if you want. If you wanted that, I mean, that'd be a good experiment to do. If you wanted to mix the 3 of them together and spot them. That actually probably wouldn't be a bad idea. Because then you'd have less of them in each, and they might not spread out as much. Actually, that's a really good idea. If we're still, you know, doing COVID chemistry next year, I might do that.
You have to think of it. Hey. Man, that will be.
So I'm going to go over. I mean, the calculations are super easy to do, but I figured I'd sort of show you what I got here. So So I spotted green twice, and they sort of split. So actually, why don't I just do that in green? That sort of makes a lot more sense. Where's my green? There it is. Right, so, oops. So green number 1, green number 2. So they separated into 2 different spots. I had blue and yellow, then my solvent front, and then the RF. So the first spot, I got 67 millimeters from the bottom for the solvent. Yellow was 42.5. And the blue is 59.5. It's going to be difficult to get much. I mean, the millimeters are so small. It's going to be hard to even get within a millimeter. But so what I did was I made my X in the middle of the spot. And if it was right between 59 and 60, I just called it 59.5. I mean, it's just way too small to try and make it 59.3 or 59.2. So, but if it wasn't directly on one of them, I just said it was in between them, which I think is fine. But you're not going to have more than 2 significant digits for the RF. I mean, we're lucky if we get 1. They spread out so much. So for number 2, solvent went 68 millimeters. Yellow went 40.5. And blue went 60. So that means the RF for the yellow for number 1 is 42.5 over 67. This is 59.5 over 67. This is over 68. That's over 68. All right. So my RF for the blue, for the number 1, is 0.63. And for the yellow, oh, sorry, wrong. The blue is 0.89. What am I doing? All right, yeah. So this is 0.63. And this is 0.89. So it makes sense. The blue runs higher than the yellow, so its retention factor should be higher. And then for the second one, the RF for the yellow is 0.60, so pretty close. And for the blue was 0.88. So not bad considering. So if you get small spots, you should be able to get fairly reproducible results. But this was using the wide paper and really small spots. Actually, my son did the spotting last night. He was my assistant. He did I don't know, he has better hands than I do or something. So he did one and I did the other one, and you could see that we got almost identical results. So we were pretty happy about that. Yeah, so the thin strip runs a lot faster. That only took about 6 or 7 minutes, really, maybe 10. And it looks awfully similar to the other one. So there is the thick strip, and there is the thin one. You can see they look pretty darn similar. Maybe I just did a lousy job spotting tonight. So on the report, in the first part, it is just basically a picture of the chromatograms of the dyes, and then the calculation of the retention factors. The second is a picture of the chromatogram of the Kool-Aid, calculation of the retention factors, and then whether you can actually identify any of the spots you see. And then the third one is basically just a picture of whatever it is you chose to do and a question about it. So that is it. So not going to be spending a whole lot of time doing calculations and plotting on the computer and everything for this one. It's fairly straightforward.
I have a question.
Yeah.
My yellow and blue are moving, but my red is still like kind of trailing.
Yeah, the red spreads out more than the other two. So yeah, I would probably just pull that out right now. And then
Even though there's still some space though, because you said like maybe like an inch in or so, or go ahead and just stop there, because they're all kind of in the same area right now. But that's like that's where my point of origin is going to be, right? Because it's the
point of origin is where you spotted it.
Yeah. Or that's the point of origin, but the
bottom.
biggest blob though is that's considered the bottom.
Right. So you'll like draw a circle around the darkest part of that blob, and then put an X in the middle of the circle.
Okay, so that's far enough then?
Oh,
then
definitely. Yeah. I mean, it's not really going to improve. It's just going to spread out more. So that's fine. Yeah.
Cool. Thank you.
But I found that was true for a lot of people that did the red spread out all over the place, and the blue and the yellow were a little bit more behaved, but not the red one. Just sort of went all over it. All right, I am finished doing my experiment. I can take off my goggles. Yeah, this is awesome. So the blue one, it's just getting darker and darker and darker on the top, while the rest of the trailing dyes catch up. So this was a huge blue smear like 20 minutes ago, and now it's all cleaned up and moved to the top. So I think I'll save it, take a picture of it as a lesson to be learned.